Welcome to the walkthrough for Biology Lab 7. In this lab we will be extracting DNA, and you're going to extract DNA from your own cheek cells. The very first thing we'll do in the lab as we'll start with a beaker that has 15 milliliters of distilled water. You're going to drink that water into your mouth. Do not swallow it, but we're going to swish it vigorously in our mouth against the cheeks. We're dislodging cheek cells from which we will later extract the DNA. And after you've swished in your mouth for at least 30 seconds, you're going to spit the water back into the beaker. And after we spit the water back into the beaker, we now have a solution of water and cells. The next thing we're going to do is we're going to add a soap solution. So 5 milliliters of soap solution added to the water and cells. And after that, we're going to add 5 milliliters of a salt solution. Once you've added these two to the solution, we'll gently swirl the solution. The soap helps break up the membranes of the cells, freeing the DNA, and stirring the solution by sloshing it or swirling it further disrupts the membranes so the DNA can be removed. We need to continue to swirl for a couple of minutes, and after we've done that, we come to what is probably the most difficult part of this lab. That is, you're going to pour alcohol on top of the soap salt cell solution. And this has to be poured very gently. Tip the beaker to the side at an angle, and very slowly, very gently pour the alcohol on top of the solution. If you pour it slowly, it won't disrupt the solution. We're going to create a layer of an interface in between the water and the alcohol, and it's at this layer that the DNA that has been freed from the cells will collect. And this is the place that we'll extract it from. And so once we've place the alcohol on top of the water, we should be able to see a layer, and if we hold it up we can see a layer. Be very careful not to stir or disrupt this layer that is there. We have to let that set now for about 5 minutes, and as it sets, the DNA will begin to float to the top of the water because the salt has made it more dense than the DNA, but it is less dense than the alcohol, and so it collects at that layer and it begins to coagulate. We should be able to see some little threads. They look like spider web threads right on that interface. And the next thing we're going to do is we're going to extract those threads of DNA that have collected there using a glass stir rod. Place the stir rod gently in the solution, place it clear to the bottom of the solution, and then turn it, twist it very, very slowly to wind the threads onto the DNA threads onto the rod. After we have the DNA threads wound onto the rod, you can't see any more of them floating in solution. You can move it around while you're turning it slowly. We will then take the test tube that has the salt solution in it, put the rod that has the DNA on it into the test tube, and spin it vigorously in the opposite direction that we were spooling it. This will dislodge the DNA into the salt solution. You may have to slide it up and down a little bit to finish dislodging it, but after it's been dislodged, we should be able to see bits of white DNA floating in the solution. You can tap it with your finger, flip it with your finger to stir it, and that you should be able to see it swirling around in there. There's one final step, and that is to prove that we have DNA, deoxyribonucleic acid, and that is we'll use methyl red. Methyl red is red at a pH of 8.2, turns yellow at a pH of 6.4 or below. And in this case, you can see that we have the yellow color indicating that we have an acid, and we have extracted our own DNA.